Nucleic acids is one of the most important macromolecules to your entire body. It is one of the most vital characteristics of life, and as far as we know, without it, life does not exist at all. It contains all the instructions necessary to build you as you. Yet, nucleic acids spent a majority of the time unappreciated. They spent a lot of time ignored by science. It wasn't until the 50s and 60s that people started to understand and get a grasp of the gravitude that was the nucleic acid. Now the question then becomes, how did science ignore this for so long? And that all deals with its basic structure. Today we're going to explore how the structure of DNA looks deceptively simple, yet allows it to do so many important and amazing things. This is Big Orange Biology. I am Mr. Jennings, and class is in session. Now, nucleic acids by definition are polymers that store, transmit, and express heredity, also known as genetic information. DNA is known as deoxyribose nucleic acid or deoxyribonucleic acid, and RNA is just ribonucleic acid. They have a special monomer known as the nucleotide. Now, the nucleotide itself is fairly a basic nucleotide. It's got a little bit more going on. It actually includes another macromolecule, but the nucleotide is pretty simple. So a nucleotide is a pentose sugar. It's either going to be ribose or deoxyribose, nitrogen-containing base, and a phosphate group. That's your typical nucleotide. Now, sometimes you'll see the term nucleosides. Nucleosides omit that phosphate group. They're just a nitrogenous base and a pentose sugar. So these are fairly common molecules. Get familiar with the look of this nucleotide, by the way. We see very similar ones coming up in the energy unit known as ATP. Now, the bases of a nucleic acid, these nitrogen-containing bases, are grouped into two major categories. Pyrimidines are known as your single ring, and purines are double ring. Now, the pyrimidines are subdivided even further into three distinct bases. You have cytosine, thymine, and uracil. And notice how I'm mentioned by a single ring. They're just one hexagonal ring seen right here. And they do contain nitrogen, hence the term nitrogen-containing bases. Purines are adenine and guanine, and they are double ring, and they are fused right here in the middle. Now, nucleotides bond in condensation reactions, much like we've discussed with every macromolecule, and their condensation reaction form what's called a phosphodiester bond. This happens between the linkage of the number 5 carbon of ribose and the number 3 carbon of the next ribose. It can also be deoxyribose. I want to point that out. And that means nucleic acids tend to grow in the what we call 5' to 3'. The 5' and 3' are on the back end here. The 5' is the carbon that sits off on its own, and the 3' is in this pivot corner right here towards the 5'. So what happens is the 5' phosphate group loses a hydrogen, and the hydroxyl group of a 3' carbon sheds itself to form water. And in the process, it creates what's called a phospho for the phosphate, and diester is the name of this bond between oxygen and carbon here. It's a very important chemical bond. This is what's going to give the nucleotides and nucleic acids in general a strong, rigid backbone. This is typically called the sugar phosphate backbone. So RNA contains ribose, and it uses adenine, cytosine, guanine, and uracil for its other pyrimidine. And it's a single-stranded molecule. Okay. DNA is deoxyribose, and it's adenine, cytosine, guanine, thymine. And it's a double strand. Pretty straightforward. Nothing too crazy. Now, they do what's called base pairing, as you may remember. A goes with T and C goes with G. So if you look here, you have hydrogen bonding going on, and the same thing's going to happen with the nitrogen. They're going to link together to form these hydrogen bonds. So the base pairs are going to be linked by these hydrogen bonds, and because there's so many hydrogen bonds in DNA and RNA, they have formed a very strong attraction. Again, remember, hydrogen bonds are still nowhere near the power of strength of a covalent bond. Because they're still nowhere near the power of strength of covalent bonds, they're actually really easy to separate. You can do it with very little difficulty, and that is very important for its function. Now, in RNA, the base pairs, like I mentioned, are A and U and C and G. Now, RNA is typically single-stranded, but in certain 3D structures, it can go into a double strand in places by folding. And that's done through the process of hydrogen bonding. Now, folding occurs by complementary base pairing, so the structure is basically going to be like how the bases are ordered, as seen here. So this is your typical single-stranded RNA, and this is more complicated tRNA. Because of this, these bases allow tRNA to actually kind of contort and create a double bonded, looks like a double-stranded situation, when in reality, all it is a single strand of RNA folded in on itself. It's kind of a pseudo double helix, double strand. Now, DNA itself is usually double-stranded. It can be found single-stranded in certain extreme situations. And because of this, is two polynucleotides are going to form a ladder with a twist that's known as the double helix. And this is your typical double-stranded DNA molecule. Pretty straightforward. Most of us are familiar with this. Now, DNA does twist in a special way. It twists on what we call right-handedness. So first off, the strands are anti-parallel, meaning one side is 5' to 3', like it's supposed to, and the other side is complementary, so this goes 3 to 5. That's necessary for the hydrogen bonds to form. Now, it's called right-handedness because the spiral, it twists towards the right thumb. If you were to hold your thumb out, you'll see it twist primarily in this direction. It is not left-handed in life. All life is a right-handed double helix. Now, DNA has information in it, and that DNA has two major functions. First off, it allows for it to code for itself. DNA can replicate based on its own instructions. That is highly critical and a very key component of DNA. Also, though, DNA has the information in it necessary to build and specify for specific RNA molecules, which in turn can be used to specify for specific amino acids, which can then go into the primary structure to build up eventually to a protein. And this whole process is called the central dogma. It's one of the most important biological chemical processes that we will ever talk about in AP biology, in any biology class whatsoever. Now, DNA's replication and transcription ultimately depends on base pairing. So, for instance, the sequence AGTCGT will be transcribed to an RNA sequence of UCAGCA. They complement each other, so wherever you see A, you put a U and a C for a G. Now, DNA replication, the entire molecule must be replicated completely so each new cell can receive its own set. And to help understand what we're talking about, I mentioned this word earlier, genome. Genome is a complete set of DNA in a living organism, and genes are mainly the sequence of DNA that actually does code for RNA, in turn meaning it codes for proteins. The genome itself does not actually code for a lot of genes, or at least a lot of the genome is non-coding regions. But very specific regions do a lot of unique functions. Now, gene expression is a term we use to talk about transcription and translation of a specific gene. So certain genes we want to express at certain times, but we don't want them all at all times because that is a waste of resources of amino acids and energy. So we do not want to do that. We try to avoid that the best we can, so we regulate the process. Now, DNA bases do reveal evolutionary relationship, as we'll explore especially in Unit 7. And that means closely related species typically share a high number of DNA, or at least a high percentage of DNA. And we can do a whole lot of stuff to actually show this. We can use it as comparison tools. Because we're able to code whole genomes, we can actually compare genomes with each other and see all the highly specific, unique regions of the non-coding DNA and see all the similarities. So in conclusion, for the longest time, scientists viewed DNA as a kind of a junk molecule, honestly. They didn't think DNA actually did that much. They were actually looking at protein for being hereditary based, because protein is so varied. However, despite only having effectively only 4 bases within them, DNA and RNA has a lot of going on. It can do so many different things because you can arrange them in different sequences. You can get totally different proteins. How you put those A, T, C's, and G's in a specific order are going to give you a specific amino acid. And because you get a specific amino acid, you're going to get a specific protein. Changing one nucleic acid, one nucleotide within that DNA molecule, you can change the entire protein. And that plays a very, very important critical role. With that said, this has been Big Orange Biology. I'm Mr. Jennings. I hope you had a great day. Thanks for listening.